Hi everyone. We are going to do regarding Mendel's experiments today. Before that, I would like to tell you what are the things related to the genetics and inheritance characteristics of the human. When we consider inherited characteristics, simply color of the skin, color of hair, and the nature of the earlobe that may be fused or freely present, or else sometimes there may be different special characteristics like some people can roll their tongue. So those are the features that we can see differently in person to person. Even country to country that will be different, like some people are having brown eyes, some people are having blue eyes, so different characters. Why this occurs? So firstly, that was found by a person called Gregory Mendel. So according to the Mendel's theories, we are going to discuss how genetic materials will be transferred from one generation to another generation. So we will see what are the experiments. So first, what Mendel has done. So he take pea plant as the sample to cross. So he has done all the experiments with pea plant because pea plant is easily can be grown in number of most of the environments, easily can be grown, and also it gives quick results. So he can see the results in a quick time period, so nearly 3 months time. So that is another advantage of that. And also it has number of features, differently characterized features. So example, taller plants and shorter plants is one feature, and round seeds and wrinkle seeds is another feature. So kind of features are there. So what he has done, so I will tell you what he has done with the tall and short plants. So he took pure breeding tall plants and pure breeding short plants. So what are pure breeding? That means that is not crossed earlier. So pure breeding tall and pure breeding short plants. So he has crossed these two plants at the beginning, tall and pure breeding short, right? So I will put the symbol. So usually genes are coupled, and two genes transmit one characteristic. So therefore I will put pure breeding capital T. I will tell how it comes later. Then shorter, simple t. So then what he has done, he crossed these two plants together. So when what he has done, he crossed this. How he crossed this? He took pollens of the taller plant and put that on stigma of the shorter plant, and he did the opposite also. He did this not only single or twice, he did this more than 1,000 times. That's how he got the good reliable result. So for all the experiment that we are doing, even in future if you are doing something, the reliability will be increased if the number of times that the experiment is high. If the number is high, you will get the reliable value. So he has done more than 1,000 times to give this result. So he crossed this, then he got, I will just draw this, he got for the first generation 100% tall. What he has done, from the first generation he crossed first generation 2+. That means that is exactly self-pollination. So previously he did cross-pollination, but cross-pollination from one feature to another feature, tall short plants crossed. But here, from the results he got, he did the self-pollination. That means capital T, simple T, and capital T, simple T, self-pollination. So the results were 3:1, 3 tall and 1 short. So I will tell you how this comes. So later on, scientists found that these features are carried by specific things called genes. So always characteristic will be transmitted through combination of two genes like here. So therefore I will tell you what happened exactly in the Mendel's experiment. So before that I will tell you how genes are categorized. So there are two things. One is called homozygous and other thing is called heterozygous. So homozygous means it has similar type of genes, gene combination. So therefore, example in above case, capital T is homozygous and simple T is homozygous. Then capital T, simple T is the heterozygous type. But they found another feature. What is that? If capital T present, capital T is called the dominant character, dominant. Even it composed, I mean, when we consider two gene combination, so even there is only one capital, that means dominant gene is there, that feature will be the dominant one, and this will be tall, because capital T is the dominant character. If the dominant character is there, result will be the dominant feature. So then this is definitely tall and this is short. So that is how the results come. So then homozygous, heterozygous genes we discussed. So how it comes now? Now you can understand how it is done by Mendel. So Mendel did pure breeding tall, so that means that is capital T, and then pure breeding shorter plants, that is simple T. So this is called first generation parents, right? Parents. Then from that later he got these gametes G1, capital T is 1G gamete type, so I will round that. Then the other one is simple T. So then the results will be capital T, simple T, that is 100% tall. You know, capital T is the prominent character, so therefore this is tall. Then what he has done, he took this capital T, simple T plants and did self-pollination among those plants. So self-pollination means capital T, simple T and capital T, simple T. So that is P2, parents, second G. Then gametes G2, capital T, simple T, capital T, simple T. He cross it like this, right? Capital T, capital T, capital T, simple T, capital T, simple T, simple T, simple T. So this was the result he got. So you can see here, this is tall. Now you understand, this is tall, here this is tall, this is tall, and this is short. This is short. So according to that, 3:1 here, 3:1 is the result he got, 3:1. So when we consider the feature that we can appear, that is called phenotype. So this is another new thing for you. So the things that you can appear from the outside, outside. So that is called phenotype. So therefore, in this case, you can see 3 tall plants are there while we are having only 1 short plant. So therefore, uh, 3:1 is the result of this. Okay, so 3:1. Then when we consider another name given is called genotype. So this is what? This is phenotype. So what is genotype? So genotypes is the gene composition. So in this case you can see capital T simple T is one gene composition, gene combination. Then capital T simple T is another gene combination, and simple T is the other. So therefore only three types: capital T, capital T, capital T simple T, simple T, simple T. So how many capital T's are there? That gene combination only one. Then capital T simple T is 2 here. Then simple T is 1. So therefore gene composition, that means this is called genotype. This is the genotype. It is 1:2:1. So in the in any exam, if they are asking to mention the genotypes, definitely have to put that under these gene compositions, or else it can make some errors. So I think you understood this. So according to Mendel's theory, he has checked only by using for all the experiments by using one feature, that is called monohybrid cross. So he has done the monohybrid cross, and the other thing is right. So other thing is, according to Mendel's theory, these genes, when that is coupled like this, it can freely segregated when it is transmitting as gametes. So here you can see, so capital T. So then freely this can be separated and transmitting to the gametes. That feature is called freely segregate. But in the next theory, that means in Morgan's theory, that is different. In that theory, that will not be freely segregate. So that is exactly called linked genes, and we will discuss regarding linked genes in our next video. So thank you for hearing. I think you got something, and join me next video. Thank you.